I didn't take a typical path with poultry science as part of my undergraduate background. I really love doing research with chickens as they fill so many different roles. There aren't problems that a single scientist has all the expertise and experience. Collaborations are really important.
Welcome to Unplugged, the official podcast of the Poultry Science Association. This is the podcast where we strip away the fluff and get real about poultry science from bench to barn. I'm your host, Andy Vance, Executive Director of the Poultry Science Association. In this week's episode, we're going to explore how poultry genetics is transformed over the past 4 decades, from early QTL studies to today's advanced genomic tools. As a global leader in chicken immunity, disease resistance, and heat stress research, Dr. Lamont is going to share what she's learned, what's next, what future scientists could keep their eyes on. She is a distinguished professor emerita at Iowa State University and is an internationally recognized expert in chicken genetics and immunity, as I mentioned, and has been studying these birds for the past 41 years. I'm really excited, Sue, for this conversation. And this is the second part in a 3-part series we have on the history and evolution of poultry science as a field and how it's contributed to the Poultry Science Association and vice versa. So let's start with this. If you go back to your first days in poultry genetics research, you're reflecting on how far we've come, the innovations, the developments, how would you describe the landscape of poultry genetics in your grad student and early faculty days relative to where we are now?
When I really started in poultry genetics, that preceded my time on the faculty at Iowa State. So that was a part of both my postdoc and my graduate studies. And I really began not so much with a focus on poultry, but on chickens as a basic biomedical model. And they have just been a fantastic discovery platform for science to look at genetics, to look at immunology. Many people don't realize how many fundamental discoveries came from looking at chickens in a very scientific manner. So that was the viewpoint that I had as I started, and that was to understand how does basic, innate, natural variation in the bird impact on important things like the immune system and the response of the birds to external stressors such as heat stress. And that is part of what has really shaped my career. And I think also the field. We've been able to go from only looking very crudely at the genetics or the genome of birds to be able to look at it in great detail, one base at a time, and looking at what a change there may have to do with the performance of the birds.
You mentioned something I've been pondering. This is, I mentioned, the second in a 3-part series. In part 1, we talked with Paul Siegel. I know you and I have talked about it. I have a great respect for Dr. Siegel and have worked with him on some things over the years. But he also mentioned that role the chicken plays in biomedical research. For people who aren't steeped in poultry genetics and maybe that relationship between poultry science and the biomedical field, what is it about the chicken and its genome that has led it to being such a useful platform for some of the biomedical research applications? Because I find that really fascinating.
Really, there are several things that have made it a great research tool. One, of course, is the great reproductive rate. So if we want to have lots of siblings in a group to study, that's quite an easy possibility working with chickens. Another is, the relatively quick generation interval in comparison to some other research animals. And we have a unique aspect of the genome being about 1/3 the size as it is in mammals. And so if we're going to study something, we have less genome that we have to analyze in order to get those answers. So if we compare it to, say, a mouse, or most typical mammals, we have about 1/3 the size of a genome in the birds. And there's been a lot of speculation about why do birds have a smaller genome, with, I think, one of the most interesting theories being that it made them lighter for flight.
Oh, yeah. Not carrying as much baggage, so to speak, I guess, genetic baggage in this case.
That's right. And it also, from a research perspective, it means that there is less, quote, junk in there and more important things that are actually controlling the biology of the bird.
Yeah, I love that. You're thinking about, if I'm thinking as a layman about the genome as the code, and you're comparing it to computer code and so on. So there's a lot less code to have to debug, so to speak. I'm thinking back to you personally, and you mentioned kind of your early interest in chickens, and you alluded to this a little bit in our first question. But when you started focusing on chickens, what was it that really drew your interest? And then how did you end up in genetics and specifically immune response in those birds?
It really started, quite early on, and that was understanding just the unique nature of birds, and then looking at those early scientific discoveries that had taken place in the immune system of birds and in the genetics of birds. And then I just had a really fortunate pathway forward and did my postdoctoral work with J. Robert Smith at the University of Massachusetts, and he gave me a lot of free rein in that particular position as a postdoc to really combine my expertise, which at that time I felt was more immunology, with his genetics expertise. And specifically at that time, he was working on a very unique genetic line of birds. We now call it the Smith line in his honor, but at the time I was there, and he had quite a sense of humor. We called it the DAM line, DAM for delayed amelanotic line, because the birds spontaneously at some point in their life would lose pigmentation in their feathers. But that was associated with a whole range of other pathologies. Some of them lost vision because the immune system was attacking the eye. Some of them developed what we'd call thyroiditis. The thyroid was being attacked. And we eventually learned through our work and that of many other researchers that it really required a combination of a specific genetics and then some type of other trigger in order to set that whole set of the disease symptoms into motion. I really credit J.R. Smith with getting me into genetics as a field.
Well, and looking at that, from the time you're working on the dam line, the Smith line, to present day, and you've been teaching now genetics for 41 plus years, you've seen genetics evolve from those classical breeding methods to now these really sophisticated molecular tools and all of the technological assistance, if you will, that we have in the process, or the way we can use those tools to better understand what it is we're working on. In that time frame, what do you consider the biggest technological leap, or maybe the leap that yielded the biggest understanding or advancements in that progression during your career?
That absolutely had to be the sequencing of the genome.
Yeah.
And along with that, developing the genetic map. Because remember, if we sequence a genome, we only have one individual sequence. But once we start talking about the genetic maps, then we're looking at diversity of the genome across hopefully a large number of animals. And that's when we can start to understand what that natural variation does in terms of changing the biology and the performance of the animals. So the genome sequence and the genetic mapping in the chicken, absolutely crucial. And interestingly, the chicken was the second animal that was sequenced, human, and then the question is, in mouse, and then the question is, what should be the first farm animal? And the rationale for the chicken is because of its distance from humans, we would get so much more scientific information from looking at a bird rather than choosing another mammal to look at initially.
Yeah, that makes a ton of sense. And obviously, as you alluded to, it's opened a lot of doors for us, in biomedical research as well as agriculture research. I'm wondering, thinking about what we know today, you kind of have those, the things you have the things you don't know, and then of course the things you don't know that you don't know. But want to focus on that second category for a minute, the things that we don't know yet. How robust is our understanding of the bird's genetics and how it influences all of these other aspects of its growth and performance and health? Like, so example, your focus on disease resistance. How much do we know now versus how much we know we don't know at this stage?
Well, I think it's becoming more and more apparent with every well-designed study that is done that there are levels of complexity in the genetics going from what is actually in the genome to what is expressed as performance in the bird. And Initially we thought once we just knew about the genes, we would have all the information.
Mm.
Well, we understand now that there are enhancers and suppressors, and the epigenetics are very important in terms of how we eventually get the variation in the genome into the actual whole bird, and that the birds or the flock's performance. So I think that's one of the most important things, is understanding that we don't have all the answers. Each time we peel one more layer off that onion of the genome, we realize that there's another layer that we didn't understand, and we're now coming to really know how very complex it is. And along with that, I think another layer of complexity that we don't know is that so many of our traits of interest, whether we're talking about growth or egg production or disease resistance or resilience to heat stress, they're traits that are controlled by many, many genes. There are just a handful of examples where it's a simple genetic control. Most of the things that we're really interested in for animal performance are quite complex in terms of the genetic control.
I'm glad you mentioned epigenetics. Share a little bit of your expertise or wisdom or that grounding in our understanding of the genetic basis of a lot of these challenges that we might deal with in chickens versus all of those other factors that might cause a gene to express itself or not express itself, a nutrition, housing, environment, and so on.
Genetics now is embedded in almost every other discipline, because how we manage the birds may impact on the expression of genes. How the birds are fed may alter the epigenome, and we will see the different performance levels from the same birds depending on how they are managed. Inspired by some work by others, we've integrated similar questions or studies into some of our heat stress work, where we looked at the impacts of what if we apply a thermal conditioning step in the eggs? What if we apply that to the eggs and then raise those chicks to be breeders and look at their offspring? So carrying that out multiple generations. So, there are so many opportunities to utilize the other disciplines within the context of improving genetics or altering the epigenetic expression of the genome in a way that'll be beneficial for the animal production.
You specifically in that kind of context have done a lot of research, your own lab, about how birds respond to disease and stress. Thinking again back over 4 decades of deep study in this area, how's our understanding of the genetic basis of disease and maybe resilience of the bird and their resistance to disease changed in the years that you've been studying this particular aspect of genetics?
I think there's been a greater recognition of generally how complex the genetic control is. But I say that with the caveat of there being some very specific diseases where there have been identified major genes that are very important. But what we need to understand will be the best way to utilize them. Just identifying the genetic control doesn't always lead us to the path which will be easy for implementation in a genetic improvement program. It's much simpler if there are only one or a small number of genes that are involved. But the other, I think, important change maybe in the paradigm has been, rather than just looking disease by disease, are there ways that we can enable the bird, the flock, to be more resilient across a whole range of pathogens that they might encounter in their lifetime? Because we don't always know what it is that are going to be the disease challenges that any particular flock might encounter. So, can we work on what we would call the innate immune system, which is the earlier stages of response to a disease? And if we work at that level, the expectation is that the resistance will actually be broader than a single virus or a single bacterium. So a lot of focus has changed to understanding the cells and the molecules and the genetic control of them for the innate immune system.
It's really fascinating, and I'm thinking about plant side of animal agriculture and all the work that's done there to make, let's say corn more resistant to various diseases or pests or, whatnot. And so you might be tempted to say, gosh, it would be great if you could create a chicken resistant to insert disease pressure here. But what you're describing is, because of the complexity of all this, maybe we're better off looking at making a more resilient, robust, resistant bird generally. Is that?
That would be the philosophy that I would have. And, corn is quite different than a chicken.
Well, sure.
We're talking monoculture, and plants have the advantage of actually having a lot of resistance genes, single genes that has been
Chickens, not so much.
not so much. We usually have a very complex situation. And that gives the bird some resilience and some balance to have that diversity of control of their different traits and resistance capabilities.
No, and I'm thinking also what you were describing earlier about the complexity of all this, right? Sometimes as a layman, you can picture it as, you just got to flip those couple of genetic switches in there and voila, something magic's going to happen. But that one switch might map to 4 or 5 other problems you could create for yourself, right?
Yes, rather than a single switch, I think of most instances as being a network of circuits. And you can take a number of different pathways to get to the same result. And that gives some measure of safety. It's kind of a fail-safe when you've got multiple ways to get to resistance.
I want to pull back a little bit and get some more of your particular story and mileage that you've accumulated along the way, because you've translated a lot of your research globally. I mentioned in your quick bio up front that USAID funded project on heat stress that you were involved in. As you've worked internationally generally, I'm curious how those efforts overseas translated or influenced your research back home at Iowa State. What component your global perspective, in other words, had on your research program in your lab?
I think that the most important thing that I and most of the researchers from the U.S. that were involved in the international work that we did was a great appreciation for 2 things. What does food security really mean? Because that's kind of an abstract concept when you live in the U.S., but when you travel and work in parts of the world where losing your small flock of chickens may have, devastating impact on the family nutrition and the poverty level of a family or a village, it then becomes very real. We also had the opportunity of almost all of the trainees, all the graduate students, postdocs that were working on the project, to also spend some time working overseas on these projects. And they all came back transformed by understanding the role of poultry, not only in highly efficient systems, which we can have in some parts of the world, but the role of poultry when the inputs are low or the feed is not very readily available, or you don't have very much space for them. So that's one of the reasons I really love doing research with chickens, is they fill so many different roles around the world, from low and middle income countries to high income countries. There's a role for them in all of those places.
Yeah, well said. And that's really powerful. So I love that you've got that perspective and shared it with us, because I'm also thinking about how poultry specifically, and I'll ask you about the genetic component of this. How is poultry genetics or geneticists or the research we're doing poised to help us meet some of this diverse basket of challenges we face globally, whether it's climate change? You mentioned food security is obviously a huge one, antibiotic resistance in livestock production. I mean, there are a number of issues we could pick. What are your thoughts on the role poultry genetics research play in helping us solve some of those or advance some of those big challenges?
It is going to just continue to expand in terms of its importance. We understand how very efficient poultry are in terms of producing high quality, economical protein and micronutrient source for the human consumers. And as we look at shrinking space in which to raise animals for food production, poultry are just very, very well suited for that. And so I see the genetic improvement being at a couple of different levels. One is the incremental improvements that we see in the higher income countries, where the system is already very efficient. The birds are very optimized for production, but we can continue to make incremental improvements, as well as looping in new breeding objectives. Consumers change in terms of what they are looking for in their food. And the breeding companies are very aware of this and really try to respond in a very good way to not only to meet, but also anticipate, where consumer preferences are going in the future. And so that's one of the areas that genetics is definitely going to be contributing to, and that is to adapt the birds to the changing consumer preferences in those parts of the world where there's enough disposable income to make those kind of preferences. In other parts of the world, it'll be to continue to improve the ability of birds to thrive and be productive in those areas, sometimes utilizing some of the local stocks that have been there, because sometimes those have been in place for hundreds to thousands of years. So they have natural selection, which has adapted them to the environment, but generally they're not very productive. Wisely using genetics to better understand the genes that are in the local birds that help them adapt to the environment, and at the same time increase the productivity of those birds, be it for meat or for egg production, that would really be a huge increase in production of poultry and in food security in those areas of the world. And genetics is going to play a big part in both of those scenarios.
Yeah, well said. Great concepts and exciting opportunities as well for folks who are listening to this. Maybe are getting involved in the field. I think tremendous opportunities. I want to dig into your personal research a little bit more. I know back in the late 90s, you were emphasizing non-MHC control of disease resistance, and I'm going to showcase my big depth of genetic terminology and for folks who are not steeped in genetic literature, the major histocompatibility complex. You might explain that for the layman's what MHC means. But you were working on this idea of non-MHC control of disease resistance. My question, in addition to what the heck is MHC and why does that matter, but have we fully tapped into the potential of non-MHC control of disease resistance yet, or do we have more work yet to be done in that arena?
We definitely have more work to be done in that arena. And early on, a lot of the work in genetics for immunology in birds really only had a couple of different genes or sets of genes that they understood and knew and could work on. Immunoglobulins, so that's antibodies, and the MHC genes. Those were ones that were accessible, even if they weren't understood very well at the time that we began. And so we not only continued work with the MHC and still do that to this day, but the recognition when more of the genomic information became available was that we could start to access the genetic information at places other than the MHC in the bird, and that was going to be absolutely essential to have a comprehensive picture of what was controlling disease resistance. So that was some of our pivot to start looking at genes that were very much involved in the innate immune response or the early immune response, some of the chemicals that are made that help to reduce viral load, or chemicals that are important to communicate from one immune cell to another one. And those were several of the things that we were looking at, characterizing, and identifying in the non-MHC areas of the chicken genome.
Thinking about the research you've done, and you're one of our most well-published members in the association, what areas of genetics are you most excited about for the next generation of researchers to explore? Thinking about, today's graduate students or junior faculty members, folks out there working in industry who are on the front end of their research careers. What gets you a little giggly about where we're going in that field?
If I was starting my career today, what I would want to jump into with both feet would be use of the technology of gene editing. That's the logical stage that we're at with our current understanding of the genome, are beginning to identify genes that are important. And we need a test system to confirm what type of outcome are you going to get if you select breeders that have a certain genetic variant in them. What's that going to look like for the future generations? And so gene editing, where we can, in a very precise manner, look at the outcome of changing or selecting or studying a single location in the genome, is really important. It's important for fundamental research, what we're going to do in a petri dish with cells in the laboratory, maybe as a high throughput screening for something that would later prove to be of value and then be translated into application. That's what makes me really excited.
Well, I love the way you set that up if you were starting your research career today. So let's look back and say if Professor Emerita Dr. Sue Lamont could give advice to graduate student or, junior faculty member Sue Lamont, what's one piece of advice that you would give yourself or to today's young researchers embarking on their career about the future of poultry genetics? What advice would you give them?
Be collaborative. There aren't many important problems that a single scientist has all the expertise and experience that's needed to solve. Our challenges are big ones. And to form collaborations, to participate in teams, bringing your own expertise in, but identifying others that are complementary to your knowledge and interests and expertise. That's kind of been my secret all the way along. And, just find outstanding people to collaborate with. It's energizing. It helps to hold you accountable, and it just allows you to really address issues that are really important and ground truth what you're doing. And one of the ways that I had the opportunity to continually do that was to have close relationships with genetics and breeding industry. Is this of value? Is this a question that you're interested in seeing answered? And sometimes that was in the form of collaborative research together. Sometimes it was simply, being in good communication to understand what would be helpful for me at a university to be doing for research, as opposed to what they had in-house capabilities to do. No reason to duplicate things. But I saw my role as fundamental research that then had the potential to be of value to go into the poultry industry in a practical manner. So for people starting out their careers, make sure you're working on something that's of value. Not everything will be successful, but you want to be able to see the potential, whether it's 2 years or 10 years down the road.
That's such powerful advice, and I would just echo that in my conversations with PSA stakeholder groups, whether that's industries or other associations or whatnot. But that's one thing when I ask them, hey, what message do you want to carry back to research community and faculty and students? And that's the one thing that I hear most consistently is, let's make sure that we're solving problems that are relevant to the industry and production. And that's really fantastic advice. All right. So the important question here as we wind up our time today together, Sue, is you've taken this step to the next chapter in Professor Emerita. Any ongoing projects or passions you're continuing to pursue? I know you joked when we talked earlier in the day that you're kind of failing at this whole Emerita thing in terms of you're still working pretty hard. So what is it that's keeping your hours occupied now in this phase of your research career?
Well, there's I think 3 things that I'm engaged with a lot yet professionally. And one is that you'll still see me out at the farm with the ISU genetic lines. I've enjoyed working with them for 40 years. That was one of the most important foundational pieces of the career that I've had, and that is the unique genetic lines of chickens that we've been able to work with. So I'm still out there working with them. There is, for any researcher, always a lot of manuscripts that need to be written up. Those always seem to lag a bit behind, and we're continuing to work on those. I think we have a lot of exciting stuff still to share with the scientific community. And the third thing that I'm working on is I'm maintaining a position on the board of several research organizations or consortia at the international level. I think that's a good way for me to give service back to the profession after a few decades in it. I think I have some maybe perspective and knowledge that's of value there. So I'll continue with most of those things probably for the foreseeable future.
That in of itself is great advice. Continuing to pay it forward, we would say, and I love that, Sue, that you're leaving us with that. Last question I'll ask you before we wrap up today. You shared with us some of the things that excite you about innovations and discoveries and technologies, but I wonder if there's one misconception about poultry science or the poultry industry that you wish you could dispel with the community at large. What's a misconception you wish more people understood?
I wish more people understood that you can jump into it at different stages in your career and that you can learn at any point in your career. I didn't take a typical path with animal science and poultry science as part of my undergraduate background. I really came into it later, but I'm a passionate learner and continued to do that along the way. I think it's turned out okay.
Yeah, you've done really well for yourself, Dr. Lamont. What a treat to talk with you. 2009 Fellow of the Poultry Science Association, has won more awards you can shake a stick at in our association, from the Zoetis Fundamental Science Award, the Evonik Award for Achievement in Poultry Science, the Helene Cecil Leadership Award, and well earned in every case because of your many, many contributions to the field of poultry science and to the Poultry Science Association as an organization, and continuing to do so and sharing your time and expertise with us today on the podcast. Friends, if you've enjoyed today's podcast, I would invite you to make sure that you subscribe to Unplugged wherever you listen to your podcast. We'll be back next week with another discussion as we move forward on all of the different aspects of poultry science and the way it's making the world a better place. Would also encourage you, membership's about the best way I know to stay involved and aware of what's happening at the association. Make sure your membership is up to date. If you haven't yet taken the opportunity to renew, do so, because it gets you our best rates, of course, on attending events like our annual meeting coming up this July in Raleigh, or our Pacific Rim Scientific Conference this October in Macau. Also, of course, you heard the reference of manuscripts. Certainly get you more favorable publishing rates at our trio of journals, Poultry Science, the Journal of Applied Poultry Research, and our newest journal, European Poultry Science. Oh, and if you are a student, of course, it's free. You gotta love that. You can learn more at poultryscience.org. Until next week, for the Poultry Science Association, I'm Andy Vance.